Cell division is the way that cells reproduce to continue life. In order to divide, cells must go through the cell cycle. The cell cycle is composed of three main parts. These are interphase, mitosis, and cytokinesis.
Interphase is the first part of the cell cycle. It accounts for 90% of the cycle and takes the most time. Interphase can be broken down into three phases. The G1 phase, where the cell prepares to replicate its genome, the S phase, where DNA synthesis occurs, and the G2 phase, where the cell prepares to divide.
G2, the chromosomes have already been replicated, and two centrosomes have been replicated by a single one. After interphase, the cell is ready to go through mitosis.
Mitosis is important since after an organism is grown, cell division still continues in order to renew and repair. Examples will be replacing cells that die from accidents or cells from the bone marrow dividing to make new blood cells.
Mitosis can be broken down into 5 stages. The first stage is prophase. In prophase, the chromatin fibers condense into observable chromosomes. The nucleolus disappears and the two sister chromatids are joined at the centromere. The mitotic spindle begins to form. This is made out of microtubules that extend out. The centrosome is surrounded by little microtubules that are called asters.
Next, in prometaphase, the nuclear envelope fragments and each chromatid have a specialized protein called a kinetochore.
Metaphase is the longest stage of mitosis. Here the chromosomes line up on the metaphase plate, which is in the middle between the two poles of the spindle. The kinetochores of the sister chromatids attach to the kinetochore microtubules. Non-kinetochore microtubules from one pole attach to those of the other pole.
Next is anaphase, the shortest stage of mitosis, which begins when the sister chromatids are pulled apart into individual chromosomes. These daughter chromosomes are pulled toward opposite ends of the cell as their kinetochore microtubules shorten. The non-kinetochore microtubules grow longer, which lengthens the cell. After this stage, the two ends of the cell have complete collections of chromosomes.
Telophase is the next stage where two daughter nuclei form. The nuclear envelopes begin to rebuild and the chromosomes become less condensed.
Following this last stage of mitosis is cytokinesis. This is the division of cytoplasm. Animal cells perform this by forming a cleavage furrow, which pinches a cell into two. Plant cells form a cell plate, which eventually becomes a new cell wall.
At the end of mitosis, the two identical daughter cells contain the same amount of genetic information that the parent cell began with. Those daughter cells are known as somatic, which are diploid cells. Their number of chromosomes can be referred to as 2n.
Meiosis is the second form of cell division. This is essential in order for life to produce offspring. It's composed of two different cycles of division, though only the first cycle involves DNA replication.
The end product of meiosis is 4 genetically unique gametes. Also, there are half as many chromosomes as in the beginning.
Two phases of cell division with meiosis are meiosis 1 and meiosis 2. Meiosis 1 is responsible for separating homologous chromosomes, and meiosis 2 for separating sister chromatids. We'll start by taking a look at meiosis 1.
Prophase 1, the stage following interphase, is where the chromosomes start to condense. This is also where crossing over takes place, which is the process by which homologous chromosomes exchange corresponding pieces of DNA molecules. As synapsis ends, the chromosomes start to gradually move apart. Spindle fibers begin to form, and the nuclear envelope starts to dissolve.
The next stage is metaphase 1. Here, chromosomes become aligned in the center of the cell according to their homologous pairs.
Following metaphase 1 is anaphase 1, which is where homologs start separating as a result of the decreasing cohesion of sister chromatid. The chromosomes move to opposite poles of the cell, while at the same time sister chromatid move towards the same pole.
Telophase 1 is the next step, and this is where two haploid cells form. Cytokinesis, or the division of cytoplasm, takes place at the same time as telophase 1 and is a factor in the formation of two haploid daughter cells. While the chromosomes continue to move to the poles, they begin to condense.
The second stage of cell division in meiosis is meiosis 2, which involves the separation of sister chromatids. Prophase 2 is the phase in which a spindle apparatus is created. Also, the chromosomes begin to move towards the metaphase 2 plate.
Metaphase 2, due to crossing over, the two sister chromatids from each chromosome are now genetically unique. The kinetochores of sister chromatids grab onto microtubules, which are coming from opposite poles.
Anaphase 2 is where the chromatids separate. They are now their own chromosomes and will move to opposite poles.
Telophase 2, the final phase of meiosis 2, is where nuclei form. Cytokinesis begins and the chromosomes start to decondense.
After meiosis is complete, one parent cell will have produced 4 genetically unique daughter cells. The 4 cells are known as gametes, which are haploid cells. Their number of chromosomes can be referred to as n.
Several key differences between mitosis and meiosis. One of the big differences is that mitosis only involves one cycle of division, whereas meiosis involves 2.
Most of the differences come from comparing the first cycle of meiosis to mitosis. Crossing over only occurs with meiosis. This is why daughter cells are not identical.
Another difference is the homologous pairs line up during metaphase 1 rather than the sister chromatids during mitosis. The homologous pairs then separate into homologs, while the sister chromatids separate into individual chromosomes.
Mitosis and meiosis serve different purposes to organisms, however, both are critically important for life to continue on.